Today I'll be presenting the results from my PhD, which was a mechanistic investigation into the acute effects of apple polyphenols on carbohydrate digestion and absorption. So our previous research demonstrated that a relatively high dose of a polyphenol-rich apple extract significantly delayed the rise in blood glucose concentrations following a high carbohydrate meal. And so we wanted to determine whether or not lower doses of the apple extract could do the same, and also examine the mechanisms by which this apple extract could be influencing carbohydrate digestion and absorption. And so we did this by firstly conducting a randomized control trial. So we had 30 participants consume either a high, medium, or low dose of the apple extract in the form of a drink, or the drink was a placebo, and these were consumed immediately before eating a high carbohydrate meal containing both starch and sucrose on 4 separate study visits. And during each study visit, frequent blood samples were taken over the course of 4 hours, and these were analysed for glucose, insulin, C-peptide, gut hormones, polyphenol metabolites, and paracetamol concentrations were also measured to assess gastric emptying rate in a subgroup. But for all participants as well, 24-hour urine samples were also collected. We then conducted in vitro work to determine the potential mechanisms of action of the apple extract, and we did this by looking at the effect of the apple extract on glucose transporters using the CACO2-TC7 cell line, as well as the effect on porcine pancreatic alpha amylase activity using a fluorescence assay. So the results from the human trial showed that all doses of the apple extract significantly reduced the early glycaemic response, as measured by the incremental area under the curve for plasma glucose between 0 and 30 minutes. And the high dose actually inhibited this by 58%. So this graph here shows the blood glucose profiles for all 4 drinks, of which the apple extract profiles were significantly different to the control drink. So Tmax was significantly delayed with the apple extract drinks being at 45 minutes versus 30 minutes for control, but the apple extract had no effect on Cmax. So similar dose-dependent treatment effects as seen in the graph here for glucose were also observed for insulin, C-peptide, and the gut hormone GIP. So taken together, these results indicate that the apple extract is having an effect on glucose absorption in that early phase. We also detected serum ferritin, which is unsurprising given the high amount of fluoridsin in this extract, but we also detected quercetin and epicatechin metabolites. Interestingly, the apple extract had no effect on gastric emptying rate, so this is suggesting that other mechanisms of action were responsible for the delay in glycemic response to the high carbohydrate meal we observed. And our in vitro results showed that instead it was likely the sum effect of the inhibition of digestive enzyme activity and intestinal glucose transport by the polyphenols in the extract that were responsible for this. And that's because our in vitro data showed that even at 0.6 milligrams of apple extract per mil, which is approximately equivalent to the low dose that was consumed in the human trial, the apple extract significantly inhibited both glucose transport and alpha amylase activity. So overall, our research supports the potential for this apple extract to be incorporated into functional food and/or beverages to aid glycemic control. Thanks for your attention.